Good morning. My name is Athanasios Litsios. I'm a postdoctoral fellow at the lab of Brett Andrews at the University of Toronto. Today I'm going to tell you a few things about how we use single snapshot images of live cells to study dynamic cellular phenomena at a scale. So in the lab we use single-cell phenomics. If you're not familiar with the term phenomics, you can understand it if you view it through the lens of other omics approaches. For example, in proteomics, our goal is to measure as many proteins as possible in a biological specimen. In metabolomics, to measure as many metabolites as possible. In phenomics, our goal is to measure as many phenotypes as possible. And because almost any measurable trait can be categorized as a phenotype, our measurements can be diverse. We can measure proteins, we can measure information about metabolites, about subcellular morphology, and so on. And we do that in single cells. So a typical single-cell phenomics pipeline entails the use of high-throughput genetics for the construction of a large number of cell lines or strains. And then we subject these strains to high-throughput live cell fluorescence microscopy, where we typically collect data from in the range of 10 or hundreds of millions of live cells. And then we use computational approaches to analyze this data and extract systems-level insights about the function of eukaryotic cells. So we are very interested in understanding the process of the cell division cycle, which very simply put is the process by which a eukaryotic cell gives birth to another cell. And also we're interested in understanding how intracellular organization emerges in eukaryotic cells. So you can understand intracellular organization as the size and shape of the different structures inside the cell, as well as the relative position to each other. And it's important to mention here that intracellular organization emerges in a new cell during the process of the cell division cycle. So these two things that we study are related to each other. So why should we care about the cell division cycle? Here you see a schematic representation of the cell division cycle of budding yeast, which is the main model organism that we use in the lab to study this process. Initially, we start with a single cell. As the cell cycle progresses, the daughter cell emerges and becomes larger and larger. At this part of the cell cycle, DNA replication is taking place. Then we have the nuclear division, and eventually we end up having two cells, a mother and a new daughter cell. And this process is essential for the reproduction of unicellular organisms, for the growth and development of multicellular ones, as well as for tissue repair and regeneration in them. And also the deregulation of the cell cycle is the cause of some highly prevalent and really devastating diseases. So I mentioned at the very beginning of my talk that we use snapshot images of live cells to study dynamic cellular processes such as the cell cycle. And by snapshot, I mean that we do not take time series measurements. And a typically snapshot image would look something like that, where we have cells from a cell cycle asynchronous population, which means that cells are in different phases of the cell cycle. And then we apply some computation to segment the cells, and then we apply further computation to extract some temporal order from these images to study cell cycle-specific phenomena. And the question now would be, why apply computation to extract temporal information instead of going directly doing time series measurements? And the simple answer is that properly done time series live cell fluorescence imaging experiments are very difficult to perform at scale. So this approach allows us to do our experiments at a really, really large scale. And at the same time, we can avoid some common pitfalls associated with time series fluorescence microscopy, such as phototoxicity and photobleaching. So you can imagine that building a new cell is quite a challenging task for a cell. So this process entails the production, the synthesis of a lot of new protein. And these newly synthesized proteins can be used, for example, as building material for the new cell, and also for regulatory purposes. And these proteins not only need to be synthesized, but also need to be distributed at different parts of the cell at the right time of the cell cycle. So the process of the cell division cycle involves quite a bit of what I like to describe as cellular logistics, which is nothing else than changes in proteome abundance and localization. And changes in proteome abundance during the cell cycle have been previously mapped in detail, but until very recently, we lacked a highly resolved proteome-level view of changes in protein concentration during the cell cycle in any organism. So in the next few slides, I'm going to tell you how we used our approach to generate the first genome-scale annotation of protein localization changes during the cell cycle of a cell. So initially, we started by constructing a yeast strain that expressed some fluorescent proteins And by these fluorescent proteins, if we would inspect these cells via fluorescence microscopy, these proteins would reveal the exact cell cycle states that the cell was when we took the snapshot image. And then we made use of the so-called yeast GFP collection. So this is a collection of approximately 4,000 yeast strains, and in each of these strains, a different native protein is fused to a green fluorescent protein, or a GFP. And via this fusion, again, if we inspect our cells with fluorescence microscopy, we can measure the abundance of the respective protein, a native protein, but also identify its localization in the cell. And I have to say that this collection of approximately 4,000 yeast strains captures approximately spans 75% of the yeast proteome and nearly 100% of the so-called visible yeast proteome. So the next step was to use systematic genetics, and specifically synthetic genetic array technology, to introduce these fluorescent mark reporters in each of the strains in the yeast GFP collection, and we ended up constructing a new library of 4,000 yeast strains, and now in each of these strains we could monitor both the abundance and localization of a native protein, but also identify the exact cell cycle states that the cell was at. Then we subjected this library in high-throughput live cell fluorescence imaging, where we collected images from a bit more than 20 million live cells. And I mentioned earlier that when we take a snapshot, cells are in different cell cycle stages. So we had to somehow automate the process of extracting cell cycle-specific information from these images. And for that reason, we developed a supervised neural network that we call CycleNet. And what CycleNet does, it examines a single cell image, and based on the fluorescent reporters that we introduced in our cells, it can tell the exact cell cycle states where a cell was when it took that image. And we found out that CycleNet performed with more than 93% accuracy. It allows us to do what we term as in silico cell cycle synchronization. So once we have grouped our cells at different cell cycle stages using CycleNet, we used a second convolutional neural network that we applied in our images that was previously developed in the lab, and it's called DeepLock. And what DeepLock does, it can identify the subcellular location of a protein in a cell with a resolution of 20 distinct subcellular localization classes. So the essence here is that by applying two distinct convolutional neural networks in single snapshot images of live cells, we could extract spatiotemporal information from them at a scale. And we found out that our pipeline worked pretty well, and I'll show you here an example. So here you see some cells. These are actually cells from images taken with microscopy. Blue denotes the cytoplasm, pink denotes the nucleus, and green the protein of interest. In this case, the protein of interest is V5, which is a transcriptional repressor. And we know that at the beginning of the cell cycle, V5 is localized at the nucleus. As the cell cycle progresses, it exits the nucleus and it goes to the cytoplasm, and it stays out of the nucleus until almost the end of the cell cycle when it goes back into the nucleus again. So these are the real dynamics of V5. In the heat map, you will see what our pipeline predicted. So x-axis here denotes cell cycle progression, and the y-axis denotes the different subcellular localization classes. And you can see that our pipeline predicts that V5 is inside, and when the color is yellow, it means that the protein is in this localization class. When it's dark, it's not in there. So our pipeline predicts that V5 is in the nucleus at the beginning of the cell cycle. As cell cycle progresses, it translocates to the cytoplasm. It stays there until the end of the cell cycle, where it goes back again into the nucleus. So which is exactly what V5 does in reality. And we found out that our pipeline could also accurately predict other well-known but way more complex cell cycle-specific changes in protein localization. And by applying this pipeline to the complete visible yeast proteome and also extracting protein abundance measurements from the very same cell images, we could generate a highly resolved spatiotemporal map of the proteome during the cell cycle. And because this study was recently published, I will not focus more on the insights that we generated. I would just like to mention that all the data generated in the study can be accessed in a user-friendly way from the cellvision.org/cellcycle, which is a dedicated web tool that we built for that purpose. And the tool is also directly searchable using human gene names for colleagues that might not be so familiar with yeast biology. This study was supervised by Brett Andrews and Charles Boone, and was performed in collaboration with Ben Grice and Oren Krauss, and also other colleagues at the University of Toronto, Harvard, Elm University, and University of Minnesota. And now I would like to take the last few minutes of my talk to discuss how can we take this further. So how can we use now potentially snapshot images of live cells to reconstruct the dynamics of subcellular structures as a function of the cell division cycle? I mentioned at the beginning of the talk that we're interested in understanding how intracellular organization emerges during the cell cycle, so how intracellular organization changes as a function of the cell cycle. And to map that, ideally, we would like to have some videos like that. So here you can see a video of a yeast cell. In dim red, you see the cytoplasm. Bright red, you see the nucleus. This cell goes to the cell division cycle. We see that the daughter cell emerges. I hope you can see that well enough. The nucleus is positioned up properly, and then we have nuclear division, and the cell division cycle ends, and it starts all over. And the nucleus is just one structure. If we want to see more structures, ideally we would have more structures on top of them. So here you see with green on the top another structure. We see actin patches, and specifically a protein that's called SAC6. And what this protein does is it stabilizes and orchestrates the actin cytoskeleton to support the morphogenesis of the daughter cell and its expansion. So while such videos would be very useful if we would have them for all structures in the cell to map the dynamics of intracellular organization, there is a secret about them. And the secret is that they are not real. Or to be more precise, they are semi-real. They are based on what we would describe possibly as biologically informed virtual cells, and I will tell you how we build them. Again, we start with high-throughput live cell fluorescence microscopy. We collect a pool of tens of millions of live cells, and then we use our neural network CycleNet to group these cells in different cell cycle stages. And then we start for each video by randomly selecting a cell from the first cell cycle stage, and then we use a score based on 3 image similarity metrics to select the next real cell in the pool from the next cell cycle stage, and we do that iteratively until we end up having selected images for each of the cell cycle positions, each of the cell cycle stages. And then we use a pre-trained deep learning model film to add motion in between these frames. So essentially, by generating a backbone of real cell images at different cell cycle stages and using generative AI, we can construct videos of yeast cells going through the cell division cycle, which can also, to our surprise, remarkably recapitulate dynamics of subcellular structures with high, I would say, biological accuracy. So we are now at the stage that we can generate such videos for the complete visible yeast proteome, so for 4,000 different proteins, and essentially that spans all major structures in a cell. And although we can see that the videos, what the videos show agrees very well with known biology, we're still looking for some ways to systematically evaluate their accuracy. And we can imagine that if we have such videos for every possible structure in the cell, integrating all this information would allow us to build a high detailed map of the biogenesis of a cell during the cell cycle. And now we're actually looking for ways to do that. So this project is a project that we lead together with Professor Raul Krishnan at the CS department of the University of Toronto, the Vector Institute for AI, in collaboration with graduate student Chen Hao Chao, and also with contributions from Sophie San, who will be presenting a poster here tomorrow. And with that, I would like to thank you. I'll be happy to take any questions.
Thank you. It's a fabulous talk. So last year, Mattis Mann published a high-resolution protein interaction in yeast. So have you classified your proteins to see whether some of the proteins are always co-localized throughout the cell cycle, some of them only co-localized for a fraction of time?
Yeah, awesome question. Because this will allow us to identify transient interactions during the cell cycle. We are now, one of the things that we're doing is, while we're analyzing these videos, we're trying to integrate more data sets into the analysis. One of these data sets is the data set that you described with the protein-protein interactions in yeast, and see what we can learn from that. I don't have an answer of what we're going to learn yet, but it's possible that we'll be able to identify, some transient cell cycle-specific interactions through this process.
So Jean-Philippe mentioned, interest in perturbations. Have you started looking at perturbations and the effect on localization?
That's an awesome question because we have generated a data set in the lab that consists of a bit more than 100 million of cells. And in this data set, what we have done, we have surveyed approximately 17 distinct subcellular structures, and we had applied genome-scale perturbations in there. So they are working on this on a separate project, but the idea is eventually to merge these things. Yeah.
Right, let's thank the speaker again.